Okay, upstream regulators. Okay, I'm not even going to ask. Say you got this example, right? We've got all these individuals. Maybe they have criminal history, maybe they don't. No criminal associations, they don't know each other, but they take orders from, they're all known to take orders from this person, right? Godfather, if you will. That's who we're trying to hit in bioinformatics. That's who I want. And a lot of times, do you think the Godfather has an alibi? Oh, heck yeah. Hey, I was at the movies. I don't know what you're talking about. I think that's the worst Italian accent I've ever done. You know, hey, and that's the thing is that when we survey a particular tissue, you've got growth factors, you've got all kinds of things in your bloodstream that affects gene expression, right? Even drugs that the people take will affect gene expression. How do we know that stuff's there? We know it is because we know it's lieutenants. Here's an example here. I never measure interleukin-6 personally, right? That in this particular example, I didn't measure interleukin-6 directly, but I see all these targets that are known to interact with it. The odds of getting that, all these targets based on my gene list, is 2 times 10 to the negative 17th. Okay? Even though I don't see the rock, I can see the waves that it leaves. And I can follow those waves back and get to the rock. Not only that, but we know a lot about this gene. That everything with an orange, it's known to induce with an orange arrow. Red indicates an increase in expression. Look, sure enough, all these things are going up. It's known to inhibit these two things. These things are going down. The only thing that doesn't follow a more active interleukin, oh, that's 1A, sorry. Interleukin 1A are these 4 here. You can see the overwhelming evidence is for a more active interleukin 1A. So not only did I see the ghost, but I know which direction he's going. And that's where the key of this stuff goes, is now we can take these upstream regulators. Oh, and here's a formula. Again, it's not complicated. Just a confidence interval. Bioinformatics at its basis is very simple statistics. Not only that, we can do this for microRNAs that we're not even measuring, chemicals, and drugs. And I tell this story a lot. I was analyzing 2 different data sets. One was a clinical trial and one wasn't. These are all older men. And the clinical trial, they can only take the clinical drug, but the control group could take, other drugs as well. When I analyzed the 2 groups together, the biggest effect I saw was Viagra. It was in the control group. You can see that. Anything you take, when I look at young people, a lot of times I could see cannabinoid responses as well. Anything that affects gene expression, I can see it. But here's what we want, and this is the big goal, is we want to get to this, right? How do we take all this information? You've seen all the, the crime movies, man. They take all, they're basically reconstructing the mafia organization. That's what we're doing for all these different things. This is lung cancer. This can be anything you want. And so in the end, this is what I want to get to. Is what is connecting all my gene expression? Where are my targets? Why does it work? What can I get at? I'm going to stop there. Let me just show you hold on a sec. Yeah, I should always know I have a big mouth, so I always go over. You can reach me. Oops, sorry. You can reach me at these places.
If you send me just this page, I'll put it up on Canvas so everybody can just go.
Okay, yeah. I used to have a web page, but something happened. Now it's dead. But I'm on Facebook. I'm even on Spotify. And I warn my high school kids, some of the lyrics are adult, but all the beats are legit. So it's just making the warning. All right. Any questions for me? Yeah.
How long, just like with one of those studies that you did, for example, the lung one, how long does that take you?
It depends. When you first do it, I've been doing this for about 15 years now, so honestly, I could probably get to that point in maybe a day, 2 days. Somebody else, it would take a lot longer, but like I said, I've been doing this a lot. So honestly, with the software that you use, it's pretty easy. And I think if you have like a biological, you know, you come from a biological base, I think it's not too bad. I think especially, and that's why I like bioengineering is, as you can see, is a lot of this stuff you can just see. Like if we can take the data and just visualize it, you know, there's so much stuff you can learn. And so that's kind of what I'm looking for, is people to take all this complicated data, you know, let's visualize it. Let's figure out what's going on. You know, put it into a way that, you know, you could explain to my kids what's going on. You know, and that's kind of the key. And a lot of bioinformaticians out there, you know, it seems like they try to make this as complicated as possible. You know, I don't program, I don't code, but, you know, I can still do what they do. And in fact, I think I could do it better because, you know, you're incorporating the biology, you're seeing it in a different way. And I think that's the key with bioinformatics. Yeah?
How much do these things cost?
Brand new, so they don't even use those anymore. That's kind of like old technology. But back in the day, they were about 600 bucks. And if you go into the microarray core here, yeah, 600 bucks to process and to buy the chip. If you go into the microarray core, there's boxes of those used chips. That's where I get them all. Every time they know I'm like going to talk to somebody because I come in the microarray core and I grab a whole handful and throw them in there. Yeah, there's thousands and thousands of dollars. And right now, what's going to happen is these microarrays are going to go away, and it's going to be RNA-seq. So they're going to sequence the actual mRNA. And every time they sequence it, they'll count it. So not only will you get levels of mRNA, but you'll actually know the exact, if there's any variants, any mutations in the mRNA. So that's kind of where it's going right now. And that's a ton of data.
Price is really high. It's like $50,000 per RNA-seq, right?
No. Right now, it's gotten a lot cheaper. I would say you could probably run your average sample for about 800 bucks. Yeah. It depends what kind of coverage you want. You know, the deeper you go, the more expensive it is. So.
And then, so it seems like in physics, you have like experimentalists and theorists, right? Theorist sits in a blackboard and does his thing, and then some experimentalist goes out there and builds something, right?
Yeah.
So that's kind of what I'm seeing going on here. Am I off base at all, or like?
I think there's a definite need for the middle ground, right? You have your biologists that a lot of them don't even want to understand this, right? You start talking about, you know, 20 million data points, and they're like, what? You know, and then you have a lot of bioinformaticians that are all computer science people, mathematicians that don't get the biology. So what you need is that middle person. I think, you know, like I came from the bench, so I'm a biologist that have incorporated the math and the computer to kind of fit that role. But that is the big need in biology right now. Who can talk, you know, who's the middleman between all this information and the biologist that I can actually do something about it? Totally. Yeah.
What's your typical day? Are you just crunching numbers the whole time, or
no? I goof off a lot. Actually, I'm actually dressed up for this. Usually, I'd be in like, you know, sandals and shorts. You know, a lot of times if I don't have anything going on, I'll just work from home. And that's a great thing about bioinformatics is I don't have to come in and like feed cells on the weekends or, you know, kill rats. And I've killed lots of rats, trust me. You know, it's nice is that, you know, I can always come back to this. Not only that, but I can, like I said, all that data's out there. If I want to know something, I'll just go get the data. And that's the cool thing about it is if something pops in my head, I have the means to go out and get my own answers. All right. Well, thank you very much. And if you're interested in this, please contact me. Was it good?
Oh, yeah, it's really good.
Okay, good. Oh, thank you. Hey, you guys can take those gene chips. I don't care. You want it? Yeah. They're collector's items probably now.
Yeah, that was awesome.
Good.
Cool. Can you send me this and I'll put it up on Canvas so that if anybody wants to contact it, they just go in and
Totally. I totally will.